Right, who's tired?
Me.
You need extra breakfast, yeah?
Yes, I need.
That's my code. That's the code for myself.
Like yesterday, even better.
Right, series cluster of differentiation. Right. So antibody-antigen interaction is the last lecture that I'll have. Hopefully by the end of the lecture, you'll have a brief understanding about antigen-antibody reaction, about cross-reactivity, affinity, avidity, and then we'll spend a bit of time on tests based on antigen-antibody interaction. Okay? So these are the things that we're going to cover. And this is the outline. Let's go into the slides. Right? So, what are, from previous lecture, what are important things for antigen-antibody interaction?
The epitope and the paratope.
Epitope and paratope, okay, what about them? How they should be?
As in they should be.
What are the criteria for epitope and paratope in order for them to be attaching to each other? They have to be complementarity towards each other, yes. You remember we said the hand and the glove?
Yes.
The 3D structure should be similar, okay? So that's one thing.
And the process between.
Yes, the molecules which are there on the epitope should be in a way that creates interaction and bonds with the paratope on the antibody.
Yes.
Yeah? So, and why that's important? So they can create hydrogen bonds, electrostatic bonds, van der Waals, okay? And be on time, guys, yeah? Because it's still 10 minutes, it's fine. Hydrophobic bonds, yeah? So these create a site where the antibody and antigen interact with each other. Okay? So it's important based on, one is the 3D structure, the other one is what molecules and elements are there. Yeah? So therefore antigen-antibody reaction is this, and the reaction is what? Reversible. Yeah? Reversible meaning what? Antibody, antigen. Attaches to this, later on goes back. Okay? It's very strong interaction, but they can be dissociated. They can be separated. Why that is important? Why do you think? Why should not antibody form a covalent link with the antigen and stick there?
Like that T cells.
These interaction is strong. Covalent is much is the strongest. It will not be separated. Okay? Why should antibody not have covalent link with the antigen?
Like we
Because
mentioned
it
in
cannot link with other things.
Well done. Yeah? Imagine I'm a university lecturer. If I get stuck in here, can I go teach elsewhere? No. Okay. Exactly. And then I have to be able to attach to other things as well. Go to different places. Yeah? So therefore it's reversible. It comes and binds and it can go. Later on it comes back again, binds to it again. Yeah? So it's a reversible and non-covalent. And if you look at from here, this is showing the structure of antigen and this is the antibody. The 3 loops that we mentioned, you remember? And it creates a surface complementarity towards each other, fixed to each other. And based on the elements over there, they have all these van der Waals, hydrogen bonds, electrostatic bonds, hydrophobic, hydrophilic properties of the molecule, they'll get stuck to each other. Yeah? So this is till now antigen-antibody interaction. And if you remember from previous lecture, all the 3D structure is very important. But one thing we can have is called cross-reactivity. Meaning what? Let's say we have anti-A antibody, right? So this is the antibody recognizing anti-A and binds to the antigen A. Yeah? Does it, if it's shared epitope, meaning this part is the same but the bottom part is different, the antibody still will come and bind to this 3D surface. Maybe not as strong as here, but still will come and bind to it. because this pocket size is the same, but this region is different. So to this we say shared epitope. Okay? Similar epitope would be if this antibody comes and binds to this, but this does not fill the whole gap of it, but they're interacted with each other. So they say shared epitope. The epitope is shared with that one. So we can have this anti-A antibody can have cross-reactivity to these antigen B or C. Okay? Meaning what? We have an antibody that based on its size and pocket, it might be able to recognize multiple different antigens. Very, like this is with the strongest one, looser with this, it can be dissociated sooner, and even the other one even sooner. So we can have this called something called cross-reactivity, meaning one antibody might be able to recognize multiple. Okay? But which one's the strongest is the one which fills up the whole gap of it. Yeah? And stronger interaction. But the same antibody might have cross-reactivity towards other antigens. Okay? Is cross-reactivity a good thing or bad thing, you think?
It's good.
Good things.
It's good because it helps us for detection.
Yes.
Sometimes it can be bad. Why? Because it might recognize our own self-antigen.
Yes, like the heart
Yeah.
in your blood cells.
Okay? Sometimes it's bad for that.
Like that.
But generally, because it's a mechanism where it can help us to actually recognize it more, it helps us for more detection. Okay? And then response. So, let's talk about affinity. Okay? Affinity means the strength of one antigen binding site, which is here, towards the antigen. So, how strong I attach to here, this is says, we call to this affinity. From this antibody, how strong it is attached to there. Okay? So, for instance, on this slide, which one do you think has a... Oh, it's written here. This, because it covers the whole area, even this antibody has cross-reactivity with that, okay? Shared epitope and all that. But because this fills all the gap and it's more matched, binds to this with higher affinity, higher strength, yeah? Compared to that one, okay? So therefore, this is quite important for antigen-antibody interaction, okay? Generally, we want molecule with higher affinity. Yeah? We want the antigen interaction with higher affinity. Why? Because it holds the antigen with higher strength. And this is a good thing that we need. So, dissociation constant, as the name means, it means like separation constants. So, how they calculate it, they calculate concentration of antigen at all times, like concentration of antibody divided by concentration of antigen antibody in the whole test tube. So what does this mean basically? The KD, it means how easy it is to separate the antibody from its antigen. You remember the two magnets I said? Okay? If the magnets are very strong, the separation constant is what? Stronger or less? I need to put more energy to separate them. Yeah? So this is one thing that you need to know about, which means, as it says here, it shows how, literally it's the reverse of affinity. Yeah? The higher the affinity, the lower the separation. Yeah? So this is what it says here. Right, shall we do a test so you can recognize avidity? Avidity means affinity was what? Antigen, one antibody with the antigen. Yeah, so
Attached together.
exactly. Strength of that.
Yeah.
Avidity means if we talk about the whole structure of the antibodies. Okay? So for instance, in this case, if we have a pentamer, yeah, the strength of that. Let's have all the guys stand up. Let's do the test. It's good for you, especially. So you antibody, okay, you can recognize this epitope. Okay, hold his shoulder. Hold the arm. Okay, you recognize the arm here, and this one here. Stand up. You're an antigen as well. And each of you can be antibodies at the same time. Hold the arm. Hold the arm. No, not you. So imagine these epitopes share each other like this. Can they be dissociated? Yes, maybe. Yeah?
Okay.
But imagine, hold your arm, you hold his, and you hold the one on the back. Okay? You hold the other one. Okay? Now, can I dissociate them easily?
No.
All of you will come together. So this is his structure. And this means avidity. Okay? So this specific antibody can recognize this, but this one can recognize some others. Or if, imagine right now, so let's make that structure. Turn towards your back. Let's say you all have same epitope. It's like 5 people, and each of them holding one person. Yeah? So this means avidity. Make sense? You get it?
Yes.
Imagine before, if he just grabs you two, I take him, you two come with him.
Yes.
But now, if you're all attached to each other, it's very difficult. You're not coming. I come with you. Yeah. You say, I want to go have breakfast. I said, I come with you. Right. So the overall strength of attachment of antigen to an antibody is called avidity. Whether it's a monomer antibody attaching to 2 antigens, or if it's a pentamer, to this we still say IgM. This is one single antibody, but it has 10 different binding sites. So the avidity of this is much stronger than that. Each individual of them, this binding of this to this, binding of that to that, is the same. Yeah? But when you have multiple antigen binding site, it's much bigger than the avidity, the strength of it is bigger than the other. Make sense? Just like what we did here. And here I think it's, is it this one or? No, it's 2 slides. So valency, meaning the location that it can bind to the antigen. So either we can have poly or multivalency, or single monovalency is Fab. You remember you said that's what is Fab? This is an antibody, yeah? One region of it is Fab. We have one Fab here, one Fab there, which I said we use it for drug. Why? Because instead of making the whole antibody, you just can use the smaller version. So Fab has how many valency? 1. It just binds to one antigen. Antibody is multi. It binds to 2. Yeah? IgM antibody, it binds to 10. It's poly, many. Okay, so this is what the valency means. And then, which is as shown here, so again, this is monovalent, binds to one side, maybe it cannot recognize the other one. Here that recognizes 2 is bivalent, or it means 2. So higher avidity, even though the strength of each of them is the same. But because numbers is important here, this has lower avidity, higher avidity. This structure here, what we've done here, it has very, very, very high avidity. Okay? Or even sometimes if we have this way, but each of them is bound to different antibodies, and what would happen? Suddenly you form like a crystal. Everyone's attached to each other. And very high strength. So here it's showing Fab, what I told you. This is showing the number of valency, how many can be attached. And so Fab is 1, IgG here in this case 1. This IgG, the second IgG, can bind to 2. This IgM up to 10. And if you can see here, the equilibrium constant, it goes higher, higher up. Meaning it's much more stronger. The avidity is much more stronger, and antigen-antibody interaction will be much more stronger. That's why complement activation uses IgM, because it helps to detect the foreign antigens. So, let's say if we have we do an experiment or a situation where we have excess antibody, okay? So we have too much antibody but not many antigen. They form little tiny small complexes. Yeah? So we have many antibody but little antigen. Okay? And to this zone, what do we call it? Free zone. Okay? But if we have the right amount, they form a big complex. Okay? And it's called immune complex. Immune complex. or also it forms a lattice as well. Okay? So if we have little antibodies, sorry, excess antibody, little antigen,
That's me.
closed zone, yes, like this scenario here, we have interaction, but which one has more strength?
The
This
second one.
Where it balances between antigen and antibody.
But with the,
And all of them form a big crystal, just literally what we did here. This is what we did here. Okay?
Yes.
And it's an immune complex. Whereas if we have little antibody with too much substrate, can they form this? No. Because as soon as the antibody sees antigen, it binds to it. It's not in equilibrium to the right amount to form the immune complex. Therefore, we have zone of antibody excess here, as shown here. It's called prozone. Immune complex is everything balanced, which is just what we did here, that experiment we did. And post zone is when we actually have extra antigen. Okay? At this here, this reaction here, that it forms a big complex, with naked eye, you can see it in the test tube. And this is, literally, majority of the experiments you do in the practical is based on here. Just this region here. The balance between antigen and antibody. And all the antibodies aligning each other and forming the immune complex. And you can see it. Okay?
And the first, so that's mean the antibody more than the.
Yes, this case.
Yeah, that's mean the immunology has a high effect. That mean will protect the body because the antibody is more than antigen.
No,
Why
it
the
is-
second one? Because it's the same.
So
But it's
this
balanced,
is,
yeah.
firstly, usually we use these terms for diagnostic tests. Okay? We don't refer them to as biological. But let's refer them as to, let's say, in a disease scenario, as you what you're trying to do. We have a case where we have little antigen, little foreign antigen, but too much antibody.
It's easy to kill.
What does this mean? Yes, the antibody will be seeing it, yeah?
Yeah.
But the point is, because there's not much of those antigens lying around, means our immune system has done a good job, not much of the foreign enemy. But if they're in the same amounts, what does this mean? This acts as a signal for the immune system. It forms a big complex. Macrophages will come and see this big complex of antibody, antigen, antibody, antigen, and complement activation can happen and all that. Okay, so it means we need extra more immune system until we have excess antibody, and there's no more antigen. Then the immune system switches down. If a case we have there's loads of antigen, okay, as soon as whatever antibody we have, they'll bind. But the immune system knows that because it's receiving the antigen still, it secretes more antibody. Yeah? So this is the disease life story. Okay? So the region where we have excess antibody, prozone, immune complex forms here, which is zone of equivalence. Everything balances between antigen and antibody. Post zone is a time where we have extra antigen, but not enough antibody. As I mentioned, immune complexes. Right, what we have here. So, as I mentioned earlier, majority of the diagnostic tests that you do in your practicals in immunology, is based on this antigen-antibody interaction and forming of this immune complex or lattice. We say immune lattice as well. We say means a macro structure. Literally, what just we made. Okay, we all be connected to each other, and you literally these antigen-antibody detections tests that we do we can use them for diagnostics for many different purposes, and I think in the 2-3 slides was in the next slide and the onward we're going to be talking about that. Regarding what we did, antibody can recognize anything. It can bind to an antigen.
Yeah.
Question: can antibody bind to an antibody?
What?
Can antibody
Yeah.
bind, so this is the antigen binding site, yeah? It binds to antigen.
I think no.
Can an antibody attach to an antibody?
No, because
No.
the affinity.
So parasite.
Like we have magnum. If we have
It
like
says
the
it will not match.
Yeah, because of like the magnum.
Okay.
So we have side, positive side and negative side.
Okay.
Like this, I think.
And there will be no receptor side because
What do you say? So they say no receptor site, like magnets say positive, negative, doesn't make sense.
like
Doesn't work.
I think it can. I think,
They can?
yes.
Why?
I don't know. Anything can happen in nature.
They can.
they can.
Antibody can recognize what?
Antibody.
Protein, peptide, carbohydrates. Antibody is made of what? So antibody can bind to antibody. So an antibody, antigen binding site, if the epitope is the right size, it might be able to attach to different parts of the antibody.
It's normal or will be disease or damage or like this?
Huh?
If the antibody attached to another antibody will make damage.
It can act as a signal, yes. That's why, do we have random antibody hanging around in our blood? We have random antibody. Remember? What is the life story of antibody? We have a B cell with a B cell receptor.
Oh, and the T cells and
An antigen comes.
yeah.
This recognizes it? No, it goes away. Another B cell comes. Does it recognize this? Yes. This specific B cell gets a signal to be differentiated into plasma cell. What does this plasma cell do? Secrete that specific antibody against that foreign antigen. Not any random antibody. Our blood is not just making random antibody. No. These cells later on will be differentiated to plasma cells. Then you will make antibody. Yeah? Makes sense? So we don't have random antibody hanging around. You're right. If we have random antibody hanging around, that would be dangerous. Yeah?
Yeah.
But now what we're talking, why I asked this question, antibody attaching antibody? It doesn't happen in our body, usually, by default. But we can have it happening in an experiment. Is that helpful? Yes, for experiments, yes. And you'll see in the future slides right now. We have an experiment where we have antibody bound onto a petri dish. Okay, so we have anti against pathogen or something. Okay, or even I want to know if I have a specific antibody with the right epitope. Okay, you can have antibody with that specific epitope and the other antibody attaching to it. This is for tests we do it. In our system, it usually should not happen. We only have antibodies if the pathogen is there. And if that B cell has got the signal to be clonal expanded, differentiated into plasma cells, yeah? Make sense? Yeah, see, immune system is very clever. More clever than us. Right, so all of our tests, diagnostic tests,
We'll take it in the practical circuit. We'll take it in practical.
you'll do it and you'll see it. You'll see this reaction. Okay, I'll show you some pictures right now.
Today?
Yeah, not today. I think later on in the semester.
Oh, okay.
Okay, but basis of all your practical is this antigen antibody interaction. Okay, and this last sentence, what does it says? It means we can have tests based on antigen antibody reaction, and we can use this to detect antigen or antibody. So we can do both. Yeah.
Yeah.
So let's go with the tests. Agglutination tests are when we have antibody and particulate antigen. So antibody as solvent, soluble, yeah?
Yes.
And if we have particulate antigens, the test would be agglutination. And what will happen, you will see an aggregation of the particles and that immune complex formation. You'll see it. Okay? So this is when we have soluble antibody, but particulate antigen.
Molecule of the antigen.
Exactly.
No.
Yeah? Solid molecule as particles,
Yeah.
not soluble.
Yeah.
But if you have them as soluble, it'll be like this, immunoprecipitation.
Okay.
Okay? And again, you will see the formation of the immune complex in this as well. Hemagglutination is literally doing agglutination using erythrocytes, red blood cells.
Okay.
And it's used, for instance, for blood typing, okay, and some blood-related clinical tests as well. Again, you will do it with, you will do it in your practical.
Practical one,
And you'll do all three.
Result.
You'll do all three.
Yeah, okay.
Okay? So let's talk about these techniques. How are we doing with the time? You tired?
Yes.
Continue. Yes, always like. You're always like, yeah, right, that's not. There's no, is there direct flight from Kish to Najaf?
No, from Ahvaz, because I'm in the
Ah.
south part of Iraq.
And how did they came from Ahvaz to here?
For my city Maysan by the car, and crosses the board by the car from Ahvaz, from the border
Oh,
of
from Ahvaz they got
yeah,
to Kish.
yeah,
Okay.
by flight.
By car?
No, by flight.
By flight.
It's impossible
Because sometimes
in the center
there's no
of the
option,
city.
you have to go by the sea, yeah? Have you ever done it, any of you?
No.
No, just play, yeah?
Yeah.
There is always some flights. Right, so let's talk about antigen antibody-based techniques. One technique which is very, very important is called Western blot. What does it, it's a process that what we can do, we can actually identify if a cell has a specific protein or not. And we need for that antigen antibody interaction. Okay, so for instance, let's say there's a tumor specific antigen. I want to know if my cells express it or not. So what do? They get the cells, okay, and using various different chemicals here, you can see HCl, peroxide, blah, 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 and different steps here, what they do, they make the cell into form of a lysate. Like literally they denature the cell, okay, get all the content out, all the protein out, and what do? Then they get rid of the RNA, get rid of the DNA because we're interested in the protein. Remain with the cell just with the protein and onto a gel, okay? Like, have you eaten jelly?
Yes.
So similar to that, but a bit more solid, okay? What do? They make little wells, and onto each of them, so patient number 1 cells, patient number 2, patient number 3, 4, 5, they put the protein lysate from the cells, and they put it in a tank. It runs a current from negative to positive, and then what will happen? The protein will go towards the gel inside the gel based on their size. The smaller one will travel faster, the bigger one will get more stuck. So therefore we can separate the proteins. Now, total protein inside the cell. How many proteins do we have inside the cell? Many. Maybe millions or even more.
Yeah.
Okay? So many different types of protein. How can I know specifically inside that single cell, inside the well of a single patient, I have that specific tumor antigen. We get the antibody against that tumor antigen, and we coat it. If it's present there, it will be stuck, and it can show us a signal, and then we can detect. And this comes like this. So let's say these are healthy individuals. These are, let's say, patients. If it shows a band with the antibody that we have, it means that the protein is there. How we can see this? Antibody attaches to the antigen. Importance of antibody-antigen interaction. So this is one technique which is used for protein detection. Okay? And also on top of that, what we can do, we can quantify how much protein we have. So for instance, these would be the 2 controls, meaning how much cell we added, but then based on the strength of these signals, we can correlate to them and say what concentration of protein is there, what concentration of protein is there, and it can give us how much of that tumor antigen cells express. Okay, so this is one test. The other test we can use for protein expression, we can use for phenotyping, is using flow cytometry. Flow cytometry is a device that we get the cells from blood, or even you can get it from tissue. Let's say you take a biopsy, you harvest the cells into single cells, you put them in a fluid, yeah, and you pass the cells in a chamber in a single line manner, and it passes through a laser based on the antibody that we use. So we can have let's say we're inside this whole room, the artery, and each of us are different cells. CD4, CD8, cytotoxic T cell, T helper cell, natural killer cell, yeah, eosinophil, basophils, each of us different cells. I want to separate CD8s. What do? I get antibody against CD8, which attaches to that. You remember before I said put iron particles with magnets? This time we put fluorescence molecule. Fluorescence molecule means what? If you give him a light, it will shine another light. Okay? So I attach green fluorescent molecule to you. Have you seen jellyfish? Picture of jellyfish with green jellyfish? Yeah?
Yes.
They have fluorescent protein. So anti-CD8 attaches to you with green fluorescence. Now I run the whole test tube in front of the machine, and it can tell us if we have CD4, CD5, whatever antibody I use. And it says like, okay, we have a component, 93% of our cells are CD4 positive and CD25 positive. Okay? And it can tell us what percentage are CD4 positive, but CD25 negative, if it's here. Make sense? This is using flow cytometry. And on this video, you can see we make cells into a single cell. We either stain it with antibody against with red fluorescent or blue fluorescent or no color. Single cells pass through the laser. We have the detector for blue, detector for red. If they're both, it'll show up there. If it's red only, if it's nothing, show up there. If it's red, on this one. Blue, here. Double positive, double signal for both, shows it there. Make sense? Why this device is very, very important is important for diagnostics. So we can have, for instance, do you remember I said HIV patients, CD4 positive? They look at the number of CD4 positives. Can you look at CD4 positive cells just using a microscope and light? No.
It's impossible.
Because T cells, T helper cell and cytotoxic T cell, looking under the microscope, they're exactly the same thing. How we can differentiate? Use antibody against CD4 or CD8 with different colors, one with blue fluorescence, one with red fluorescence, and we can separate them. So this is important for flow cytometry. Also, we have a flow cytometry device that it puts a camera in front of the laser. It does not just tell you if it has blue signal or red signal or purple signal or whatever. It takes a picture as well. And it's called image stream, and this is it. So this is showing single cells, and each of them, these channels with different antibodies. You can see, for instance, let's say this is anti-CD4. It's showing higher expression of CD4 in the corner, yeah, on this cell, on this one here, on this one there. Whereas on this one, it's showing for nucleus, it shows inside of it. Antibody against the nuclear content, it shows inside, but it takes a picture of it as well. So this is another device. Right. Rick? It's 10:47. No, wait, let's summarize, then we go for it. So, antigen-antibody interaction. This is the lecture, last lecture we have. What we talked, we said, what are important for antigen-antibody interaction. One is the 3D structure. And the molecules which are there is very, very important because it creates that big interaction through van der Waals, van der Waals bonds, through hydrogen bonds, electrostatic bonds, and hydrophobic properties, making the antigen and the antibody binding strongly to each other. Okay? And affinity is the total antigen strength of antigen and antibody interaction of a single antibody. A single antibody usually having two antigen binding sites, so it means binding to these same epitope. But let's say a single antibody of IgM molecule can bind to 10 epitopes. So the strength of them might be the same, affinity would be, for each of them might be the same, but the total binding, which is called avidity, is much, much stronger. With the case of IgM, having how many antigen binding sites? 10 of one single epitope. Yeah? Then we talked about that dissociation constant, meaning how much you have to put energy to dissociate the antigen and antibody. Yeah? So the stronger dissociation constant, yeah? The higher dissociation constant, meaning what? Binding is? Yes. Yeah, binding is weak. Right, then what did we talk about? What's next? Cross-reactivity we talked, you remember? We said sometimes the antibody can have cross-reactivity with different epitopes, and we talked about why that could be good or not. Then we talked about polyvalency, monovalency, meaning monovalency in case of what? Fab, remember? Fab, fragment antigen binding, just points to one antigen. Antibody is bivalent, usually, like IgA. IgA as a dimer, sorry, as a mono. That's bivalent. IgM is how many? 10, it has 10. IgM. 10, so it's multivalent. Yeah, so you can bind together. What else did we talk? Then we talked about antigen-antibody interactions. We said agglutination is when you have soluble antibody, but antigen is as particulate and solid. Yeah, and then it forms the immune complex. Yeah, and then hemagglutination is when you have the red blood cells, and we do the agglutination on them. Immunoprecipitation is when you have soluble antibody and soluble antigen, and precipitation forms. Okay, and that do you remember the diagram? When you have excess antibody, what is it called?
Excess.
Excess. So one was prozone, one was postzone.
Yes.
Yeah.
Equivalence.
Equivalence was in the middle. What happens in the equivalence? What does it mean?
They're equal number
Of antigen and antibody, and it forms a big immune complex or lattice. Yeah, big macrostructure, just like what we do. And then, what is the prozone? Is when you have excess antibody, and then we have the immune complex. Then we have postzone. Postzone is excess antigen. Yeah. Then we started talking about antigen-antibody interaction. We talked about Western blot, which is for protein concentration, and finding if we have a specific protein within a cell of a patient. Okay, and for that we have to use antibody against that protein. Then we talked about flow cytometry. Yeah, which to the antibody we attach them fluorescence molecule, different colors, purple, green, red, yellow, whatever, and the machine detects these colors and it can help us with detection. What time is it? 10:51. Be back at 11:01. 10 minutes. And then we'll continue.